Okay, we're going to finish up our chemistry lectures talking about biochemistry. Biochemistry is the study of the chemistry of biological sciences, or the study of life, the study of the chemistry of life. So, first of all, biological molecules, there are three major biological molecules. And what I mean by major, these are also called macronutrients in the parlance of nutrition. And they are molecules that have to be consumed in larger quantities, gram quantities, as compared to the milligram or microgram quantities for the micronutrients. And the first of these larger macronutrients is carbohydrate. And carbohydrate, the name tells you sort of what it's all about. It's the combination of, it's a hydrated carbon compound. So carbohydrate is a polyhydroxyl aldehyde, which you don't have to remember. It's also a ketone, which what you need to remember is it's the water of carbon, and its simplest formula is CH2O. It's used to store energy in the form of starch or glycogen. It's an energy source for cellular function, glucose being the molecule everybody's heard of, but there are other molecules that also form energy sources. And it's also a structural component of many plants and animals. Cellulose, which is the principal component of plant cell walls, and chitin, which is a very important component of the wall of the outer structure of insects and other arthropods, are just two examples of structural molecules and structural components that are formed out of carbohydrate. There are monosaccharides, mono meaning one, so one glucose molecule or one molecule, disaccharide, which are two carbohydrate molecules joined together, and polysaccharides, which are multiple or a large number, by definition more than two carbohydrate molecules linked together. Now, a monosaccharide, as I said, contains a single component, a simple sugar. Glucose and fructose are the only simple sugars that occur in nature. Others have to be created. Glucose is the building block for long-chain carbohydrates. And here are some examples of monosaccharides. The first two, glucose and fructose, are naturally occurring. And you can see, if you look at the compound at the top of each of the molecules, you can see the difference is where the double bond O is compared to where the OH is. And then the other one, mannose, can be formed by exposing fructose to a base, and it will rearrange and form what's called mannose. Now here, the first set of structures are called linear or straight train. The second set are the rings that occur, and you can see that you can form a 6-member ring with glucose. 5 of the 6 members are carbons, 1 is an oxygen, and you have a CH2OH side chain. Or you can have a 5-member chain, as with fructose, and you can see that one of those again is oxygen, but you have 2 side chains of carbon, hydrogen, and oxygen. Disaccharides are carbohydrates that consist of 2 subunits linked together, and they're attached with an oxygen molecule. 3 disaccharides are important in our diet: lactose, which is milk sugar, maltose, and sucrose, which is table sugar. So here's lactose. And lactose, as you see, you can study these a little bit and look at the difference between the various ones. Here's maltose. And then lastly, here's sucrose, or table sugar. You can see that they look very similar, but they're not exactly the same. For example, if you look at sucrose, it's a glucose combined with a fructose. But if you look at lactose and maltose, those are both glucose molecules linked together. And the difference between maltose and lactose is how they're arranged. You can notice it says alpha D, and that's 2 alpha Ds in the middle there in maltose, and it's 1 alpha D in the lactose. Now, people that have lactose deficiency or lactase deficiency have an enzyme, do not have the enzyme that breaks this oxygen molecule, and that causes lactose intolerance. So here's another diagram looking at the 3 essential disaccharides. The top is table sugar again. The middle is lactose. And the bottom is maltose. You can see the bonding of sucrose and maltose are very similar, and lactose has different bonding. Now, polysaccharides are simply long chains of sugars that are how most sugars exist, or how most carbohydrates exist in nature. So, for example, our essential sources of carbohydrate, wheat, rice, rye, corn, etc., the sugars exist as long chains called polysaccharides. Starch is just a type of polysaccharide that contains both straight and branch chains. Glycogen is a storage form of polysaccharide that is found only in animals, and it's branch chains only. So here's a starch molecule, and here's a glycogen molecule. You can see that in some cases they look very similar. Other cases they might look quite a bit different. Now the second macromolecule is protein. Now protein is fundamentally different than carbohydrate. Protein is long chains of amino acids that represent one of the most important molecules in the body. Now, it's long chains of amino acids instead of long chains of carbohydrate molecules like glucose. There are actually 20 different amino acids, and these are the building blocks of protein. They have an amine group on one end and an acid group on the other end, and that's why they're called amino acids. An essential amino acid is an amino acid that has to be supplied in the diet that the body cannot make for itself. And so here are examples of a couple of amino acids, and what you see here, these are actually generic amino acids. The little side chain R here is what determines the type of amino acid. On the left of each of these is the amine group, and on the right is the acid group, and in the middle is what tells the different types of amino acids. And then when they get linked together, as in the bottom diagram, then you have what's called a peptide bond that links them together, links the amine of one group to the acid of another group. And this peptide bond causes, you have multiple peptide bonds when you have a protein. If you have just 2 amino acids linked together, it's called a dipeptide. Here's another example of various amino acids, and what you can see is that the difference in various amino acids are what is represented in the R group. So for example, lysine, which is the left side middle group, has a very long 4-chain carbon. That represents the R group there. Whereas tryptophan has a complex ring structure that represents the R group in that amino acid, and so forth. The peptide bond is the bond that holds the 2 amino acids together. A dipeptide is a combination of 2 amino acids, and a polypeptide is just another name for a protein. It's a polymer of many amino acids linked together. So here shows the peptide bond linking the C group, carbon group from the acid, to the N group of the amine. And when this happens, water is expelled, and you have this linkage that occurs. And this linkage can form very long chains, such as shown in this diagram on the right. Proteins have multiple functions. One of the most important functions is the enzyme function. And an enzyme is a catalyst that helps chemical reactions proceed in a forward direction. They bind to molecules for transportation. For example, hemoglobin binds to oxygen and transports oxygen throughout the body. They also transport molecules across membranes. They are structural molecules. Many of the structures, like hair and nails, are pure protein. They produce motion. Muscle fibers are primarily protein. And then they regulate cellular activity and metabolism. There are actually two types of hormones. There are protein or amine hormones, and then there are cholesterol hormones. The amine hormones are, for example, an example of an amine hormone is the thyroid hormone, which regulates cellular metabolism. And then they lastly protect the body against disease. Antibodies, which fight against disease, are protein molecules. Now the third macromolecule is the lipid. The lipid is composed of fatty acids. It's the acid core with long chains of hydrocarbons on the side. Glycerol is a 3-carbon alcohol that forms the base for the lipids to form. And fats are lipids that are solids at room temperatures, whereas oils are lipids that are liquid at room temperature. So this is glycerol, and the important thing about glycerol is these 3 OH side chains here, and the long chains of hydrocarbons link up here. And so here's a long chain hydrocarbon with a carboxylic acid group. So this is a essential fatty acid. It's an acid because it has this acid on the end, and it's fatty acid because it has a long hydrocarbon chain. And then these, at this point here, link to glycerin, and each OH links up right along. Glycerin's right here. This O is from the OH on the glycerin, and then these double bond O's are from the O here, and then the OH here and an H from here forms water, and then you have these 3 long side chains. Now, these show no double bonds, and then you have one little double bond here. This is a saturated fatty acid because if it didn't have this double bond, if it was all single bonds, it would be called a saturated fatty acid, meaning that it has no double bonds. If it's an unsaturated fatty acid, it means it has double bonds and it's not saturated with hydrogen atoms. So saturated means that all the possible binding sites are bound by hydrogen and there are no double bonds present. Unsaturated means that some of the binding sites are available for binding, which means that one or more double bonds are present. A steroid is a fat that has 4 rings. And here's an example. And cholesterol is such a fatty acid, or such a fat. And you have 4 rings, and generally you'll have different kinds of side chains that determine which type of steroid this is. Now, cellular processes that occur chemically include the function of enzymes, which catalyze and facilitate biochemical reactions. And enzymes work by what's known as the lock and key model. And what happens is that the reactants, you remember those are on the left side of the equation, the enzyme holds these reactants in an ideal position and orientation such that they can undergo a chemical reaction most easily. This is just an example of a 3-dimensional structure of an enzyme. And the dark blue represents the reactants being held in a certain configuration and close together. Here's the example of the lock and key model. What you see is that they only fit in this enzyme a certain way, and when they fit in the enzyme that way, this facilitates the production of the product AB.